the first cell over here, we can see that we're using Python within Databricks to run this cell. We have other languages that can be accessed through this button in Databricks. So in here using Python, we are importing the CSV file, the diabetes.csv file that we manually uploaded to Databricks, and we're storing it in a PIMA data frame. And then what we're doing is we're printing this data frame. So the first thing we do is we click on this run button to be able to run this and see the output. So we can see that the output of the PIMA data frame shows that we have 9 columns and 768 rows. We can also see the details of what each column is about and a snapshot of what it contains. So this concludes setting up the PIMA Indians notebook. The next step we want to do is we want to run and set up the BRFSS notebook. This notebook uses BRFSS data, and to view the data and documentation of this, you can go and click on this link over here. Or again, you can go to the Google Docs link that is provided in the Moodle, and in here you can see a folder called Codebooks. And then you can go inside of the codebooks and you can see the BRFSS codebook that contains all the data, dictionary for the variables within the BRFSS dataset. So the first thing we want to do to be able to run this dataset is to connect our cluster. So again, I'm going to click on my cluster, and this means that it successfully got attached to this notebook. Now I can run each cell. So first I'm going to click on here and run this. What this is doing is basically importing the zip file from a website. So unlike the PIMA Indians dataset, where we needed to manually import the diabetes.csv file, this code automatically gets the dataset for the BRFSS data from this URL link. So this means that this cell was successfully run. Now in cell 5, what we want to do is convert the CSV file with R and save it into temp. We can see that this takes around 2 minutes to run. So we're going to go ahead and run it. And we can also see that this cell is run using R. Now we can see that this cell has successfully run. Now the final step, which is cell 6, is to copy the CSV file that we just downloaded and store it in the DBFS file system. So if we run this file, we can see that this final part of this code prints all the files that are found within the file store/tables within Databricks. So what we expect to find in this cell is 2 data sets. One is the BRFSS dataset that we just uploaded, and 2 is the diabetes CSV file that we previously manually uploaded. And this is what we see over here. So that means that we successfully uploaded both. Finally, we want to work on the NYTS notebook. This is very similar to the BRFSS notebook. So again, this notebook uses NYTS data. You can get the data and documentation using this link or using the folder within the Google Drive link that I previously talked about. The first thing we want to do is again attach our running cluster. And then very similar processes are done here as in the BRFSS notebook. So for example, in cell 3, we're getting the data set using the online URL link, and then we run the cell over here. And then we go to cell 5, we have to install OpenPyXL to convert the Excel file that we get from the URL website to CSV using Python. And then the final step is to convert the Excel file to CSV. Now this final cell takes a long time, so I previously ran this and it took 13 minutes. And finally, after converting the Excel file to CSV, we want to do the same step and copy the CSV file to the DBFS file system. And then finally, we print all the files within the system. And now we can see the BRFSS CSV file, the diabetes CSV file, and now we just added the NYTS 2020 CSV file.